All right, we left off talking about the small intestine and mechanical digestion. So segmentation was new. That was squeezing the tube of toothpaste, the closed tube of toothpaste, breaking up larger particles. And that's only in response to pressure, distension in the small intestine. And peristalsis does occur, but there's a few sequences that line up. And when those sequences line up, we framed it as the migrating motility complex. So you have peristalsis over a foot or 30 centimeters. It stops, and then another section later that picks up the peristalsis. It's not one long wave-like movement through the 2 meters of small intestine. But overall, these act to break down the physical shape of food into smaller components, which is good. Because in order to break down food into the smallest components, enzymes need access. You don't want to have one giant clump of food that prevents pancreatic juice or gastric juice from accessing that foodstuff in the middle. You need fully mixed liquid chyme as it moves through the small intestine. So now, with this physical makeup broken down and with a slurry in the small intestine, we'll look at how it's absorbed. And this should parallel the chemical aspects of digestion lab that you did a week and a half, 2 weeks ago? I can't remember. But the enzymatic digestion and then subsequent absorption is largely at the villi and microvilli of the small intestine. So the surface area drastically increased, even though we have those ridges that sometimes span the full circumference. The ridges themselves in the small intestine are lined with finger-like projections called villi. And you can see that here. These are the ridges that we looked at the gross macroscopic level. And then villi on these ridges, which look like a fuzzy coat of carpet on top of the ridges. Now we don't stop there. The villi themselves have their own carpeting. The microvilli are microscopic tufts of hair that line the individual villi and increase surface area, another order of magnitude. So villi may be a millimeter in length, microvilli a micrometer in length, 1,000-fold different. Milli to micro, 1,000 times different in size. When we talk about absorption on the next slide, we will be at the microvilli level. The microvilli are where the capillaries, the lacteals, the absorption through cells occurs. So that's where we're at, microvilli level. This is to provide context. Now, in this state, in contact with this massive surface area, the pancreatic juice, the intestinal juice, creates this loose mixed chyme. So all the substances required for digestion are mixed equally throughout the fluid. The fluid is the vehicle that carries the foodstuff and some of the enzymes into contact with the microvilli. And that's really important because not all of the enzymes that break down food are in the juice. Some of them are in the membranes themselves. The final enzymes are in the membranes themselves. We refer to those as brush border enzymes. The brush border is the fuzzy layer which, even under a microscope, when you can't see the microvilli, they're all clustered together. It makes the border of the individual villi look fuzzy. The brush border are the collective name for the microvilli as they span and line the villi. In that brush border, in the membranes of those cells, there are enzymes that will act on food that come into contact with those cells. And I'll show you a depiction of that coming up. Just like there are receptors in the membrane or transporters in the membrane, These are enzymes in the membrane. They're facing out into the lumen. Food washes over them. The chyme washes over them. And any undigested particles will be worked on, will be cleaved, will be broken down by those brush border enzymes. Now the food that we are curious about are all the major macronutrients and then nucleic acids, which we don't often touch on too much. But in digesting these macronutrients, let's consider briefly where we're coming from. Carbohydrates, you've seen in lab, and we acknowledge that salivary amylase starts the process of breaking down carbohydrates. You observe that process in the test tube in lab in an acidic environment with carbohydrate, with the enzyme. Digestion should occur after an hour. Sometimes not even after an hour, depending on if you mix the substance properly. It doesn't take an hour for food to get from your mouth through the esophagus to your stomach. So salivary amylase doesn't really have an hour to work before it is inactivated. And it is inactivated by the acid in the stomach. There is a little bit of breakdown of carbohydrates by salivary amylase, but it turns off pretty quickly. So something else has to pick up the torch. Proteins, again in lab, you saw pepsin, gastric release of pepsin in an acidic pH with the appropriate substrates, and the enzyme will break down protein. Pepsin will start breakdown, and it's continued by the release of pancreatic peptidases, trypsin, chymotrypsin, elastase, carboxylase, there's a couple of them. Overall, these are a class of enzymes that will break apart peptide bonds. The same exact process that you saw outlined in lab. This happens in the stomach, and it happens throughout the small intestine. Most of the proteins are broken down by the time they reach the brush border. Some of them are not. For lipids, we mentioned briefly that there are lipases in saliva, but lipid breakdown doesn't happen in the mouth. Lipid breakdown starts in the acidic environment of the stomach, but it really only gets going after it's emulsified. Remember the role of bile? You needed to include bile to suspend lipid in that watery substance in order for the enzymes to have access. Otherwise, it all clumps together, and the enzymes will only act on the outer surface of that glob. When it's emulsified, you break up the glob into many small particles, giving access to those lipases. So the majority of lipid breakdown occurs in the small intestine through pancreatic enzymes. Now we digest all the food we bring in. We even digest any of the enzymes that stop working, or if there's dead cells in the intestines that are sloughed off, we'll break those down too. We want to recycle all the components of our body as best we can. I want to take a quick tangential video break to review the processes of transport and absorption, just a quick 4 or 5 minutes, because when we talk about absorption, we review these processes, and I assume you know them. And when we get to renal physiology, it requires that you have a good understanding of diffusion, facilitated transport, active transport, etc. So we're going to do a very quick 5-minute review of those processes.
60 to 100 trillion cells of the human body are surrounded by extracellular fluid containing substances needed by the cell, such as oxygen, ions, nutrient sources of energy, and ingredients for cellular functions. This fluid is also the location for disposal of wastes and carbon dioxide. All of these substances need to be transported across the cell membrane. Recall that passive processes include diffusion, facilitated diffusion, and osmosis. These processes occur as a result of concentration gradients of the substance, thereby requiring no energy source to move the substance from one side of the cell membrane to another. Active processes utilize energy to move substances across the membrane, generally from areas of lower concentration to areas of higher concentration. Active transport involves protein transporters known as pumps. Other active processes use membranous transport vesicles. There are two types of active transport, primary and secondary. Primary active transport requires energy derived from ATP. An important primary active transport mechanism involves the movement of sodium and potassium across the plasma membrane using a sodium-potassium pump. 3 sodium ions in the cytosol attach to binding sites in the pump. This triggers ATP to bind to the pump and split into ADP and a single phosphate. The released phosphate attaches to the pump, causing it to change shape and open to the opposite side of the membrane. The affinity of the sodium binding sites decreases, causing the release of the sodium ions into the extracellular fluid. The changed shape of the pump now increases the affinity of the potassium binding sites, and 2 potassium ions are drawn into the pump and attached. This action releases the phosphate, and the pump returns to its original shape. The potassium ions are expelled into the cytosol as sodium ions enter the pump, and the process continues. Secondary active transport uses the energy released when one substance moves down its concentration gradient to move another substance, such as glucose, against its electrochemical gradient. The driving force is primarily the movement of sodium ions from areas of high concentration to areas of lower concentration. When 2 substances are transported in the same direction by secondary active transport, the protein transporter is known as a symporter. Glucose molecules are co-transported with sodium ions from the extracellular fluid into the cell. The occupation of the glucose binding site causes the symporter to change shape, exposing both substances to the cytosol. When the sodium ions move down their electrochemical gradient into the cytosol, glucose is released into the cytosol against its concentration gradient. Antiporters are protein transporters that move solutes in opposite directions. For example, hydrogen ions are transported out of the cell against their concentration gradient as sodium ions move through the same antiporters down their electrochemical gradient into the cell. This movement of hydrogen ions is important in the regulation of blood pH in the kidneys. When larger particles or greater quantities of solutes need to be transported across the plasma membrane, another active process is utilized. Vesicular transport involves the formation of solute-containing spheres known as vesicles. The types of vesicular transport are identified by the direction of the movement. Vesicular transport from the extracellular fluid into the cell is termed endocytosis. Exocytosis refers to vesicular transport out of the cell. All cells secrete materials such as hormones, digestive enzymes, or neurotransmitters via exocytosis. A membrane-enclosed secretory vesicle in the cytosol fuses with the cell's plasma membrane, and its contents are released into the extracellular fluid. Endocytosis occurs in 3 ways: phagocytosis,
clever ways to get things where they don't want to go. And we'll see all of them. We'll see all of them on this figure, except for primary active transport. We're starting easy. If you were bored by that, that's great. That means you knew it. But you need to know those processes and understand what a symporter is, what an antiporter is. Now, where it's not so critical, and in the renal physiology section where it is. So we're starting easy with carbohydrate, which is largely facilitated diffusion. Facilitated diffusion meaning diffusion with a pore, with a protein that opens up a hole. Not just passive diffusion through membrane, but there is a specific transporter or opening sized for the carbohydrate specifically, facilitated diffusion. So we already acknowledged that there's little breakdown by salivary amylase for carbohydrate. There is breakdown by pancreatic amylase that leaves these small fragments. Small fragments. And this one is only 4 glucose units long. It's a small polysaccharide, meaning it has many individual saccharides. And usually poly is the prefix you use for 4+. Monosaccharide, disaccharide, trisaccharide, polysaccharide for everything else. So a small polysaccharide is left after the pancreatic carbohydrate enzymes break down incoming food. How do we get this into the body? We can't take in a 4-saccharided strand into the body. We have to break it down into individual units. So one of the brush border enzymes in the membrane of the microvilli is called alpha dextrinase. And it cleaves one at a time. I thought I did all of them. Imagine that it cleaved one at a time all of these individual glucoses to be released and then transported through facilitated diffusion into the cell. For anything that's fairly long, 4 saccharides or more, alpha dextrinase breaks them down. Now, some of the small particles left over after pancreatic amylase has a crack at carbohydrate are sugars, disaccharides. So sucrose is shown here. This is table sugar, glucose and fructose. Maltose, 2 glucoses. And lactose, the enemy of some, glucose and galactose. And you don't really have to memorize the compositions, but these are different pairings of monosaccharides. And each of these has their own individual enzyme, again, in the brush border cells. The membrane of the cells in the microvilli have enzymes poking out that specifically break down these dipeptides. So sucrase breaks down sucrose, maltase breaks down maltose, lactase breaks down lactose, and boom, they all go through. Facilitated diffusion. For carbohydrates, it really is that simple. The large majority of breakdown into individual carbohydrate units, monosaccharides, occurs via this mechanism. There are small bits left over after the pancreatic juice has a crack at the incoming food. The individual carbohydrates are released in this way. One thing that I want to point out before, 2 things before we're done. The uptake of these carbohydrate units depend on the type. Fructose really easily moves in. Fructose doesn't even need a transporter to move in. Fructose moves into the cell and then out of the cell very quickly. Glucose and galactose need something else. They are actively transported. They don't just move freely with their concentration gradient. And the type of transport we just saw in that last video, it's not ATP dependent. ATP dependent is primary active transport. This relies on the movement of sodium. Sodium moving in will bring either glucose or galactose in along with it. This is an example of secondary active transport like we just saw. So part of the reason why we have carbohydrate and sodium in a sports drink. You want to get sugar into your body so you can work better? Why not leverage this mechanism? Why not also add fructose so you can go around that mechanism? We do that. Lastly, no, I think that was it. Oh, I wanted to talk briefly about lactose intolerance. Some individuals don't have this enzyme, lactase. My wife is one of them, unfortunately. So any lactase in the small intestines can be broken down. You can't get the individual carbohydrates into the body. The disaccharide sits around in the small intestine and is fermented by gut bacteria. Just like sugar is fermented by yeast to make beer, it's fermented in the small intestine to make hydrogen gas, which doesn't stay in the intestines for long and must escape. Maybe some of you have some experience with that, I'm not sure. We'll come back, talk about protein absorption, fat absorption, finish off the large intestine in class on Thursday.